Okay, so good evening, everyone. We'll be starting with the discussion week 8 of the course Experimental Biotechnology by Professor Vishal Trivedi. And in this week, we'll be discussing about different techniques that are used to culture the organisms in vitro. Okay, so in vitro means inside the lab. So the techniques and how we actually grow cells inside our in vitro system to replicate the in vivo conditions. Okay, so for that, let's start our lecture. So here is the first question: what is the major difference between a prokaryotic and a eukaryotic cell based on their DNA? So here are the options. Please go through it. Okay, any more answers? Okay, please read the options carefully. Okay, so in prokaryotes, as the name suggests, okay, so karyo means nucleus. It's a Greek term, so in other words, it means nucleus. Okay, and pro means primitive. Okay, and in eukaryotes, U means advanced. Okay, so whenever we are talking about the prokaryotic and the eukaryotic organisms, we have to first differentiate based upon their DNA or the chromosomes that they have. Okay, so in case of prokaryotes, we have two types of DNA present. First is the chromosomal DNA, okay, or the nuclear DNA that we call it, and the other one is the extra chromosomal DNA. The chromosomal or nuclear DNA is circular in case of prokaryotes, although this is also circular. This the nuclear DNA is large and secondly codes for all the important genes to survive for the survival of the organism, being it metabolic, structural, or any other enzymes or proteins that are needed. Okay, in case of extra chromosomal DNA, it is not present in every prokaryotic organism. Okay, but it helps the organism to outstand the other organisms. Okay, for example, the antibiotic resistance, the basic example that is being provided by the extra chromosomal DNA or the plasmid DNA. Okay, so it doesn't have any necessity to the organism, but it provides an additional advantage. That's it. Okay, so this is the case with the prokaryotic DNA. But in case of eukaryotes, we have only one major, like it also have two types of DNA. Okay, first is the nuclear DNA, and second one is the organelle DNA. Okay, so nuclear DNA present in the nucleus, it's generally linear. Okay, it's mostly linear, and it is divided into different chromosomes. So the number of chromosomes vary in different organisms. But in case of prokaryotes, if we say how many chromosomes of the genomic DNA is present in a prokaryote, that will be only one. Okay, because that is the major nuclear DNA that is present. But in case of eukaryotes, the chromosome depends upon the organisms. Okay, for example, fruit fly, the Drosophila, has n is equal to 4. That is 4 pairs of chromosomes it has. Okay, in case of humans, we have 23 pairs of chromosomes, or rather than 23, we have 22 plus X or Y. Okay, so this is the case. Then coming to the organelle DNA. So organelle DNA is basically derived from the prokaryotic genomes. So it is circular, and same as case of the plasmid DNA, uh, it is necessity of the organelle. Okay, it is not the case that it might be present or might not be present, but it is present in the organelles like mitochondria or the chloroplast. Okay, so this provides some genes that are present encoded by those organelle itself. So coming back to the question again. So the first option is prokaryotic cells have linear DNA while eukaryotic cells have circular DNA. So this is totally wrong. The option 2, prokaryotic cells have multiple chromosomes while eukaryotic cells have a single chromosome. It's the opposite case. Okay, so the prokaryotic cell would have single chromosome, but eukaryotic cells have multiple chromosomes. So this option is also wrong. Prokaryotic cells lack DNA while eukaryotic cells have DNA in form of plasmid. So this is also wrong, as we have discussed. Now, the last option, prokaryotic cells have a single circular DNA molecule while eukaryotic cells have multiple linear chromosomes. So this option is correct. Okay, so the correct answer is option number 4. And this is the typical examples of a prokaryotic and a eukaryotic cells. So prokaryotic cells, as you can see, this is the DNA. So the nucleus is not well defined here, and the DNA is present in combination with the cytoplasm itself. Okay, with attached to different proteins that are bound here, and that stabilizes the DNA. And in case of plants and animals, so these are eukaryotic cells, and here you can see, as in the image, this is the defined nucleus, and here this black region, as if you can visualize, this is the DNA wala part. Here in the plants also, this is the uh, sorry, this is the nucleus region, and this is the DNA wala region. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. Carbohydrates in a growth medium is used for which purpose in the cells? Is it energy generation, nucleotide synthesis, amino acid synthesis, or protein synthesis? What can be the correct answer? Okay. Yes, the correct answer is option number one. Okay. So whenever we see the term energy generation in any terms, okay, so the first thing that must come in our mind is carbohydrates. And out of the carbohydrates, the main constituent that is used in most of the mediums, that is glucose. Okay, so this is majorly used for energy generation in all the media components, being it plant, animal, bacteria, anything. Okay, and this, so if you guys remember the part of the metabolism, the first thing that is used for energy generation is carbohydrates. Then moving on to proteins, followed by fats, and the last one comes is nucleic acids. Okay, so this is the energy retrieval process. So first, the energy is retrieved from the carbohydrates, followed by proteins, followed by fats. Okay, so this is the path that is taken by our body for energy generation. Okay, and this is the different components that are basically used for different sources. Okay, so for example, if we have to see the protein source in a medium, so or a nitrogen source, that means any form of crude protein, so generally we use peptones. So this is basically the partially denatured protein that can be in simpler forms and can be used by the bacteria or the cells that we are growing. Then comes the growth factors. So growth factors are generally used for mammalian cell cultures or the plant cell cultures, but not the bacterial cell culture, because bacteria can directly utilize the carbohydrate source and grow. But in case of animals and plants, we need certain and more growth factors that help us to grow these cells. Okay, so in that case, we need blood, serum. So these provides growth factors that are present inside our body. Then comes the energy sources. So here the major energy sources carbohydrates or sugars. Then the buffer salts for maintaining the pH, because the pH of the organism is maintained. So we need buffer salts also. Then comes the mineral salts and metals. This is for maintaining the structural integrity of the cell, because certain metals are also needed there. Okay. Then we have selective agents that help us to grow only the specific cells but not the contaminations. For example, in animal cell culture, the major contamination is microbes. So we use certain antibiotics so that the microbes do not grow in that culture. Okay. So then the indicator dye, for example, we use phenol red. Okay, then the gelling agents like agar or alginate or gelatin that we use mostly in case of the bacterial cell culture, but not bacterial and plant cell culture, but not in case of the animal cell culture. Okay, so any doubts in this particular question? Okay, if not, then we'll move on to the next question. So dash is added to the DMEM mammalian cell culture media as a buffering agent. Sodium bicarbonate, ammonium sulfate, sodium phosphate, or potassium phosphate. What can be the answer? Okay, so DMEM is a medium that is widely used for growing the mammalian cells. Okay, so uh, it's widely used for growing the mammalian cells inside the in vitro conditions, and its full form is uh, Delbeco modified eagle medium. So it is widely used and it has much more components that we'll see later. So yes, someone said sodium bicarbonate. Okay. Any more answers? Okay. So tell me one thing. Uh, if we are culturing a mammalian cell in vitro, so what will be the buffering uh, things that are used that can be used to maintain the pH of this culture medium? Consider the buffer systems that are present in our blood. So in our blood, the buffering agents that are present mainly is, first is the carbon dioxide and the bicarbonate ions. Okay. Second is the albumin and various proteins that are involved in the blood. Okay, so these are also helping uh, maintaining the blood. Just a second. So yes, so this helps majorly in buffering inside our blood. Okay, so helps in the buffering of the through the blood throughout the body. So whenever we are growing any mammalian cell in vitro, we use sodium bicarbonate, that is a form of the bicarbonate ions, so that it helps in buffering of our buffering in that petri dish. Okay, so that's why sodium bicarbonate is widely used as a buffering agent in any of the mammalian cell culture medium. So DMEM is widely used for growing the mammalian cells. Okay. So anybody raised hand, I guess. So do you have any doubt? Okay. So yes, in this case we use sodium bicarbonate, and also while culturing the mammalian cells, we have certain amount of CO2 that is supplied to them, because our body cells are also growing in a CO2 rich environment. Okay, so nearly 5% CO2 is pumped so that the cells can grow easily. Okay, so any doubts in this particular question or related to mammalian cell culture? I've covered the basics only. If not, then we'll move on to the next question. Which of the following is an example of defined synthetic medium? Nutrient agar, tryptic soy broth, LB broth, and minimal salts medium. Okay. Any more options? So the defined or synthetic medium is the one, okay, whose constituents we know. So all the constituents that are present in that particular medium are known. So that is why it is called as a defined or a synthetic medium. Okay. So out of the following options, the nutrient agar have a component called as yeast extract. Okay. So in this case, we do not know the exact composition of what is does this extract contain. Okay, so same is the case with LB and the tryptic soy broth. So there are components called as peptones. The exact composition of these peptones we don't know. We only say that these are degraded or the simpler form of the proteins that will help us is as a nitrogen source. But the exact composition is not known. Okay, so that is why these three do not come under defined medium or the synthetic medium. The minimal salts medium have different salts that are required for the microbes to grow. Okay, so the composition of this minimal salts medium we know, and the composition is mentioned here. So this is M9 medium basically, and there are different minimal salts medium that have different compositions. So here we have the different forms of ammonium salts, magnesium, calcium salts, the EDTA, iron, zinc, manganese salts. So these salts help the microorganism to grow. These are required for their growth. Okay, and the technical meaning of the minimal salts medium is that you are providing minimal substrates for the microorganisms to grow. Okay, so that is why it is the minimal salts medium. That is why it is called as a minimal salts medium. So any doubt in this case? Okay, if not, then we'll move on to the next question. Dash is added to the DMEM mammalian cell culture medium as a growth factor. So just now we discussed what can be the answer. Okay. So yes, as we just discussed few questions back, so yeast extract is added in a bacterial cell culture. Okay, DMEM is the name of the medium. Okay, so it contains different components. So this will also be not the answer. Sodium bicarbonate is a buffering agent. Then comes the fetal bovine serum. So yes, the serum will provide all the growth factors that are needed for the mammalian cells to grow. Okay, so the correct answer is fetal bovine serum. Now moving on to the next question. Mammalian cell culture media is filtered and not autoclaved because these are the options. What can be the correct answer? Okay, any more answers? So, what happens in case of a mammalian cell culture media like DMEM? We have components like fetal bovine serum that basically provides the growth factor. So, whenever we autoclave a media or we do not autoclave a mammalian cell culture media, so in that case, what happens? This FBS, if we autoclave, this will get degraded. Okay, so that is why, because it's heat sensitive basically, and in case of autoclaving we'll go till 121 degrees Celsius. So all the components except for the media components, okay, the salts can remain there, the glucose can remain there, but the serum components will not be get degraded at that temperature. Okay, so the correct answer here is option number 2. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. The pH meter is not used for checking the pH of the DMEM media because what can be the correct answer? Okay, any more answers? Okay, perfect. Okay. So what happens whenever we are preparing the DMEM media? So first we dissolve the media aut- in autoclaved water. Okay. Then this media, the this is this we get in a powdered format. Okay. And then what this contains basically is the buffering agent, indicator, and the salts. Now, then in addition, we add FBS. So after preparing this media, then we add FBS and then we filter it. Okay, and generally we do not ch- uh, use a pH meter here to check the pH because the media components that we have might block the pH probe. Okay, because we have different particulate matter that is present inside the media. So this is the major reason that why we do not use the pH to check the pH of the this particular DMA media. So the correct answer is option number one. Okay, there are chances that media can get contaminated, but that is not the correct reason for me. So any doubts in this question? Okay, if not, then we move on to the next question. Okay, so what happens? Uh, uh, you know the basics of the pH meter, right? So it has a bulb, and here is the a, a meter that basically measures the hydrogen ion concentration inside the particular like solution that we have. Okay, so this is a bulb kind of structure, and here we have a glass electrode. So now, when we put this particular thing in a media that is basically a mammalian cell culture media, so in that case, we have different particulate components like FBS. Okay, so if you see that only small pores are present here so that the liquid containing the ions can go through it. Okay, so if we have FBS kind of structure, then it will go and get stuck in these pores. So what will happen? The probe bulb or the electrode that we are using will get clogged easily and will not get efficient readings from the medium. Okay, so that will not be possible to check the pH of that particular media. Okay, so Is the explanation correct right now? Okay. So let's move on to the next question. Why is serum heat inactivated before putting in cell culture media? So here we are talking about the serum, like we added FBS, right? So why it is heat inactivated before putting in cell culture media? So heat inactivation means that if you are adding anything that can be that can have different roles. So first we inactivate them using heat, and then we add into certain media. So why it is done? That's the question. Okay. Any more answers? Okay. So the correct answer is option number three. Okay. So what happens sometimes whenever we need a serum free media? Okay. So in that case, what we do is we use the serum and heat inactivate it. Okay, sorry, not in case of serum free media. In case where we need to study the different aspects of the sorry, in certain cases we only need the actual components of the media. For example, we have only the DMEM without FBS. So FBS is not there, but we only need the background of the media to study different morphological cells. So in that case, what we do, we inactivate the serum from that, and then we add into the cell culture media, so that we can inactivate the components of the serum that are there, so that they will not be interfering in those studies. Okay. Because we have different growth factors here that that helps different cell types to grow. So that's why the correct answer is option number three. Okay, so let's move on to the next question. Dash media is used for production of baculovirus in lepidopteran cell lines. So what happens is, when we are adding serum, so that doesn't mean only the growth factors are added. Okay, so if we have to inactivate some components of the serum that are not needed for the cells to grow, then only we use the heat inactivation. But because we need the serum to grow the cells, because only the growth factors are not needed, right? It also has certain components that helps, like the proteins, the modified proteins also present, that helps the cells to grow. So in that case, we use the in heat and activation process of the serum. Okay. So the next question is, dash media is used for production of baculovirus in lepidopteran cell lines. What can be the correct answer? So the correct answer is option number two, that is TC100 medium. So this medium is specific for the growth of Lepidopteran cell line. That's why here it is mentioned that uh, it is basically used for bacteriovirus production specifically. Okay, so this is a fact. So that's why others are. This is growth. This is for mammalian cell culture, and this is for the uh, insect cell culture. Okay, and for inks medium, this is also a insect medium, insect growth medium. So this was a factual question. Okay. So if you have to produce baculovirus in Lepidopteran cell lines, then TC100 medium will be more preferable, because in general in these cases we would need high quantity of the viruses to be produced. So that is why we need a sufficient system so that we can produce different uh, like for the high amount of production of those particular viruses. Okay. So let's move on to the next question. Which of the following is a characteristic feature of eukaryotic cells but not prokaryotic cells? The presence of ribosomes, nucleus, cell wall, or circular DNA? Yes, the correct answer is nucleus. As I discussed in the first question, the characteristic feature of the eukaryotes is a well-defined nucleus. Okay, and prokaryotes do not have a well-defined nucleus. So the difference is majorly the nucleus part. So correct answer is option number two. Let's move on to the next question. Dash allows for the mobility of prokaryotic cells. Flagella, phospholipid, cytoplasm, or lipopolysaccharide? Yes, the correct answer is flagella. So it particularly helps in mobility. That means movement of the prokaryotic cell. Okay, and this is the so there are two basically extensions of the prokaryotic cells. One is the pili, and the other is the flagella. Okay, and there is one more that is called as fimbriae. So out of these, pili helps in some cases it helps in movement, but in other cases it helps in basically conjugation. So that is one form of the reproduction in prokaryotic cells. The other one is flagella. So if a prokaryotic cell needs to move from one place to another or swim, so in that case, the flagella is an appendage of the prokaryotic cell that helps in its movement. And the third one is fimbriae. So fimbriae helps in attachment to the substratum. Okay, so this is the case. So these three are the basically extremities of the prokaryotic cell. Okay, so any doubt in this particular question? Okay, so let's move on to the next question. The region between the inner layer and the outer layer of a gram negative prokaryotic cell membrane is called as. Okay, so it's periplasmic space. Okay, and again, if we can see here, so what happens in this case, if we see, this is the outer membrane. Okay, this is in yellow, that is the outer membrane. Okay, then comes the cell wall, that is shown in red, and then comes the again the wall, that is called as the plasma membrane. So the region between the plasma membrane and the outer membrane is called as the the region outside the cell wall and the bit under the outer membrane is called as the periplasmic space. Okay, so this is, sorry, so this is the outer membrane, this is the plasma membrane, and here comes the cell wall. So the region between these two layers is called as the periplasmic space. And majorly the degrading enzymes, the complex degrading enzymes are present in the periplasmic space, and here only small proteins are present here. Okay, so this provides also an added advantage to the some of the bacterial cells. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. What is the role of phenol red present in the mammalian cell culture medium? Okay, so yes, phenol red is a pH indicator. If it's a neutral pH, so in that case it will be red in color. Okay, so it is help in monitoring the pH of the system. So we are not using a pH meter to measure the pH of the mammalian cell culture. That's why we add phenol red in this case. And different any deviation from the neutral pH that we use in our mammalian system. So in case of the our body itself, the optimum pH, what is the optimum pH in our case? What of the blood that we generally use as a neutral pH? Yes. So it's a range between 7.2 to 7.4. Okay, so this is considered as the optimum pH of our body. So this pH is only used for the growth of the mammalian cells also. Okay, so if the pH is more than 7.4, then the media turns pink. That means it's, yeah, that's what I'm explaining right now. So if the pH turns basic, that means it turns pink. And if the pH is less than 7.4, in that case it will turn yellow. Okay, so acidic, it turns yellow, it basic, it turns pink. Okay, so here the correct answer is option number 2. So any doubts in this particular question? Okay, if not, then we'll move on to the next question. In gram-negative bacteria, porins are present in which membrane? Outer membrane, periplasmic space, cytoplasm, or phospholipid layer? Okay. So yes, the correct answer is option number 1. That is the outer membrane. So what happens, as I just showed, that this is the outer membrane, the cell wall, the plasma membrane. So the outer membrane is fully permeable. So most of the porins or the pores that are present in the outer membrane, that allows free flow of most of the molecules. Okay, that are present in the environment. The major permeation starts from the cell wall. So cell wall also has pores that are specific. So it allows only those particles or those substances that have different pores or different proteins to transport the molecules inside are present in the cell wall. Okay, so the first defense comes from the cell wall in case of the gram-negative bacteria. Okay, so the porins that are present are majorly present in the outer membrane. Okay, so porins are basically proteins that have a shape of a pore. Okay, so these form like a pore kind of structure inside the membrane, and they will allow only for example, they also have specificity. So for example, they can allow either water molecule. So the porins that allow only water molecule are called as aquaporins. These are present in mammalian cells also. Okay, and there are different porins that allow only certain ions. For example, they are specific for magnesium, copper, and calcium. Okay, so these are basically pores that are present in the membrane and allow only specific molecules to pass through it. Okay, there are some porins that can be general. Okay, they can allow many substances to pass through them. So any more doubts in this question? Okay, so let's move on to the next question. Bacterial cell wall is composed of cellulose, peptidoglycan, chitin, or exopolysaccharides. Okay, so peptidoglycan is present in the bacterial cell wall. What is the plant cell wall made of? Yes, it's cellulose. And fungal cell wall? Yes. Okay, so let's move on to the next question. During bacterial media preparation, antibiotics should be ideally added to cell culture media. What can be the correct option? So yes, in case of bacterial media preparation, what we generally do is either we take a flask. For example, this is a conical flask that we use for culturing the bacteria, and then we add water and directly add media to it. Okay, in some cases uh, we can heat it, then we can autoclave it again, but there is no point in doing that because when we are autoclaving, the heat is generated in the same amount. So if we can directly autoclave it, then it is already fine. Okay, so we add the media powder. Okay, we can add either the media or the direct components. Okay, the different components of the media, and then directly autoclave it, we'll get a clean liquid. This will be the media, and now we have to add an antibiotic for its preparation. Then what we do is we cool it a bit, then we add the antibiotics. Okay, so we cool down first and then we add the antibiotics, and then the temperature of this cool down must be less than 50 degrees. That is the optimum temperature at which the antibiotics are basically sustained and they are not degraded basically. Okay, so the correct answer is option number 3. And if we add it before autoclaving, then it will be degraded. So there is no point in adding the antibiotic before autoclaving. Okay, so any doubts in this particular question? Okay. So what happens whenever we prepare a bacterial culture media? So we have two kinds of preparations. So first is all the components are there in a powder form that you directly add, and for example there is defined amount that is added for a defined volume of the media that we are going to prepare. Okay, and the other one is there are different components, you add it and mix it with the media, also with the water. Okay, so what the initial times people used to do, first they used to heat the media and then they are used to autoclave it. Okay, but in case of autoclaving we already reaching a temperature of 121 degrees celsius, so there is no point in heating. That is an additional step. So to reduce that, what we generally do nowadays is we add the media components in the water and then we directly autoclave it. It will get boiled as well as sterilize itself. Okay, now after autoclaving, what we do is we cool down a bit, so that if we have to culture anything, then we have to first cool down it. Okay, now here is the case when, if there can be two cases. First, if we are using the broth means liquid media. So in that case, if it is cooled down, then we can store it in 4 degrees Celsius. That is not an issue. But if we are using a solidified media, that means we are adding agar to it. So we have to plate that. Okay, so the media is liquefied, but agar that solidifies very quickly. So if the its melting temperature is around 37 degrees, so after reaching below 37 it starts solidifying itself. Okay, so what we do is, in this case, first we try to cool it down just to 50 degrees celsius or less than that, a bit less than that, so that now we can add the antibiotic and plate it in a petri plate. So just after reaching it to the 50 degrees Celsius mark, then we add the antibiotic and we directly add to the petri plate, so that we can get a uniform colony or uniform plate. Okay, so this is the case. In case of a broth, whenever we are utilizing it for giving a culture, we generally add the antibiotic just before that. Okay, so we add the antibiotic, then we add the culture. Okay, so this is the proper way that we use for utilizing the bacterial cell culture. Okay, so let's move on to the next question. While preparing a mammalian cell culture media, a student makes mistake and ends with getting contamination in the media as given below. Identify the mistake. What can be the correct option? Okay, any other answers? Okay, can you explain why? Why second option? Yes. So, if we can see, the average microbial size that we are having is greater than 0.22 micron. Okay, so this microbe, that can be bacteria, protozoa, any other non unwanted stuff that can bring contamination to the mammalian cell culture. So that is greater than 0.22 micrometer. So if we are using a filter that have a pore size of greater than 0.22 micrometer, so in that case our bacterial media, sorry, our cell culture media might have some contamination. Okay, that can be in the form of microbes or other particulate matter. Okay, so that is why we generally use a filter that have a pore size of 0.22 micrometer, and that will able to prevent any of these microbes or particulate matter to enter into the media. So only the media components, being it FBS, bicarbonate, or any other growth factor that is present in the media, to be in the present medium itself. Okay, so the correct answer is option number two. And if we are using a point 0.5 0.45 micrometer filter, so in that case all the bacterial cells can enter into the media and cause contamination. Okay, so any doubts in this particular question? Actually, mammalian cell culture is sensitive towards anything. Okay, so if we uh, like have any of the if we do not disinfect or sterilize the surface itself, it can cause contamination to the uh, mammalian cell culture. Okay, and even a small fungal or bacterial colony is growing inside your the tissue culture room, that would cause a hazard to the cell culture area. Okay, all the culture plates can be destroyed in a minute. But in case of bacterial or plant cell culture, culture, we do not have that much of the restrictions. Okay, so that's why mammalian cell culture is a really sensitive technique, and we have a separate room for both the things. Okay, so let's move on to the next question. In a certain study performed by a group of students on an unknown cell type yielded the following fraction ABCD. Four steps of centrifugation at 600 G, 10,000 G, 1 lakh G, and 3 lakh 20,000 G. The different organelles present in ABCD is, this would be C. Right. Okay. We got the first answer as 3. Yes. So what happens? This is the case of the centrifugation, differential centrifugation. Okay, so in case of centrifugation, the like electrophoresis. Okay, it also separates the molecules on the basis of their molecular mass. Okay, so in a well or a tube, if we have different molecular mass components, then the most heavier ones would be located in the last row. Then comes the less than that, followed by the more lighter weight. So the lightest would be on the top of the tube, and the heaviest would be on the base of the tube. Okay. For example, we have the cesium chloride gradient centrifugation. So in that case also, we perform first the gradient. In that case, what happens? The different gradients are performed, and then we add our sample, and based upon the molecular mass, it will get separated or segregated in a particular gradient. Okay, so based upon that, we will have different sedimentation coefficients for the different organelles that we are separating. Okay, so this is the so this is the explanation that I was giving you about. Okay, so the more molecular weight components will be present at the bottom, and the lighter ones will be present at the top. Okay, so now if we see, the soluble proteins would be the bottom ones. Okay, followed by the microsome, the viruses, DNA, and then comes the uh, ribosomes. Okay, so the first thing that actually separates is the heaviest one. Okay, so here in this case at 600 G. So if you remember a cell diagram, the most amount that is present in the cell is the nucleus. So it is actually more more in amount in case of a cell. Okay, so it will be separated first at the lower centrifugal force. Okay, so the here the G is basically the centrifugal force that we are applying. Okay, so at the smallest centrifugal force, the most weight uh, component will be separated first. So here comes the nucleus. Okay, followed by the mitochondria, then the ribosomes, and at the last, because plasma membrane will be separated in the form of different small particles, because it is a amphipathic molecule, because it has both hydrophobic and hydrophilic components. So it forms various small soluble molecules. So it will be separated in the highest force. Okay, so that is why if we see in the increasing order, the first is the nucleus, that is the highest, then comes the mitochondria, then the ribosomes, and then the plasmid. Okay, so any doubts in this question? This separation is based purely upon the molecular weight of the different organelles. Okay, okay, if not, then we'll move on to the next question. The most appropriate use of detergents in the process of differential centrifugation of cells is, what can be the correct answer? So, whenever we are extracting any protein, organelle, DNA from the cells, first, so in case of a bacterial cell, I am talking about first. This is the cell wall, the cell membrane, and here is the cytoplasm, nucleus, and different components. Okay, so first we have to break open the cell wall using different enzymes. Okay, so if we have the peptidoglycan, so we have lysozyme for that. So it will break open the cell wall. Then we have to lyse the plasma membrane. For that, what we'll do, because the plasma membrane is made up of fatty acids, phospholipids basically. So phospholipids can be break open using with the help of detergents. Okay, so detergents will help in breaking open the plasma membrane, because yes, SGS is one of the detergents that we generally use for breaking open the plasma membrane. So these basically remove the interaction between different phospholipid layers that are there, the phospholipid molecules, and will separate open this membrane, break this membrane basically, and that will help us to remove all the components in the cytoplasm. Okay, so it basically helps to lyse the plasma membrane and extract the cellular organelles. So correct answer is option number 3. Okay, so any doubts in this question? If not, then we'll move on to the next question. Which of these properties can be determined using a cell counter? The exact density of an individual cell, cell surface properties, quantitative growth kinetics, or size of the DNA. What can be the correct answer? Okay, any more answers? Okay, so basically, the correct answer is option number 3. And here is a typical cell counter that is used in a laboratory. Okay, so this is a slide kind of thing, and here we have two components. So first, what we do is we for this is the aerial view of the slide. So this is the glass slide, and in the middle we have certain box-like structures that are basically the area where we put our sample to count the cells. Okay, and first we place a cover slip before it, and then we add the sample, the diluted sample basically. And this is a typical mesh kind of structure present in that particular space. Okay, so now we have here four quadrants. So this is the first quadrant, this is the second, this is the third, and this is the fourth. Okay, and in case of each quadrant we have 16 boxes. Okay, so 4 by 4, that is 16 boxes. So now what we do is we count the number of cells in each of the quadrant, and then we take a mean out of it. So that basically tells us the number of cells that are present in that much ml or microliter of the medium that we have put on this particular slide. Okay, and this is how the cells look like under a microscope. Okay, so this is the number of cells, and we generally take only those cells that are present inside the boxes. So generally we avoid taking the cells that are on the edge of the or the lines that are present around the boxes. Okay, so now, and this is generally used for the viability cell testing also, and we use a dye called that Evans blue. So that basically tells us the cells that are viable and that are actually alive in case of a medium. Okay, so we check how many cells are there, and based upon that we prepare our dilutions to put on a second plate. Okay, so this is how we use a counter to determine the number of cells or the cell density that is present in a plate. Okay, so it is used to check the quantitative growth kinetics of the cells. Okay, so any doubts in this particular question? Okay, if not, then we'll move on to the next question. So, bubble formation is prevented in a mammalian cell culture systems because it causes shear stress, changes color of the media, changes temperature of the media, or causes contamination. Okay, any more answers? So, basically, what is the source of this bubble formation? Okay, so generally, bubbles are formed from the froth, and froth is basically the source basically is due to the proteins present in the medium. Okay, so whenever we shake a protein, that causes frothing. Okay, that is aggregation of the protein basically. Okay, so in this case, what happens whenever we use different proteins in a cell culture medium, we avoid making froth, although the froth comes. Okay, but we have to minimize that frothing because the frothing causes s- shear stress to the cells. Okay, that will cause a pressure on the cells and they can lyse. Okay, so that is why we avoid bubble formation in case of a mammalian cell culture system. And as you can see that the bacterial and the plant cells have a cell wall, so those are more stable in a culture condition, but in case of a mammalian cell the cell wall is not present. They only have the plasma membrane, so they are more sensitive to any shear or osmotic stress that is present in the medium. So we avoid the shear stress by preventing the bubble formation in a medium. So the correct answer is option number 1. Okay, so is the explanation fine? It can be a source of contamination, but it's mostly uh, leads to the shear stress to the cells. Okay, then let's move on to the next question. So confluency means, what can be the correct option? Okay, any more answers? So yes, the correct answer is option number D. And here is the typical explanation to explain the confluency. So what happens whenever confluency is generally used a term in case of a mammalian cell culture system. Okay. So whenever we culture or passage a cell, so that means subculturing a cell line. So in that case, first we add certain cells to a plate. Okay, so this is the typical example where we have added some of the cells to a plate. Now we allow them to settle to adhere to the culture plate, and then what happens here? You can see the number of cells are less as compared to the whole plate. Okay, so for example, if you consider this as a plate, so this is not as a plate, this is a picture of a part of a plate. So in that case, the area covered has very less amount of cells. So in this case, the confluency is less. Okay. Now, if we allow them to grow, then it covers the surface of the cell culture plate. So that means it increases its confluency with time. Okay. Now, this is the case where the full culture plate is filled with cells. So here this case is called as 100% confluent cells. That means no surface is left for the cells to grow, and all the surface area of the cell culture plate is being utilized by the cells itself. Okay, so here this is the first day after plating the cells, followed by the last day. That can be two, that depends upon the number of cells that you have put in a plate. So here the confluency increases from this particular case to this particular case. Okay, here the cells considered can be 70% confluent. Here it might be 50% confluent or less than that. Okay, so that depends upon the time period that we have left the plate or the cells to grow. Okay, so confluency basically means the number of cells that covers the surface of the culture dish. Okay, so any doubts in this particular question? Okay, if not, then we'll move on to the next question. What will be the color of phenol red in a cell culture media if there is a bacterial contamination? It turns blue, it remains red, it turns yellow, or it turns pink. What can be the correct answer? Okay. Any more answers? So, as I said a few questions back, there are three forms of phenol red. Okay, in case of neutral conditions, that is a general body pH, it is red in color. In case of acidic, it will turn yellow, and in case of basic, it will turn pink. Okay, so in the bacterial contamination wala case, the media turns pink. Okay, and here is the explanation. So for example, there is an infectious agent. So what happens, this increases the turbidity of the medium and a sharp change. Okay, so sharp change means what happens when there is a contamination, that is a bacterial contamination. So in that case, the bacterial cells basically lyse the mammalian cells, leading to release of all the cytoplasm components in the media. So that changes the pH of the media from 7.2 to 7.4 to nearly 8. Okay, as we can see in the image itself, this is the typical pH 8, and this is the exact color how a bacterial contamination looks like in a mammalian cell culture media. Okay, this is a normal red color, and this is the acidic color that is due to the like if the cell density is really high in case of a media, as I showed you, 100% component cells, so in that case the media appears yellow. That means it has high metabolic activity. Okay, so this is the case in different cases that are present in a mammalian cell culture media. Okay, so any doubts in this particular question? Okay, so if no doubts, then we'll close the session for today. And if you guys have any doubts in the presentation or in the any of the components of the week, then you can ask. Okay, then we can close the session for today. Thank you guys for joining.